Hi, I'm Holtom. After finishing up my second year at the University of York, I worked on an 8-week BSPP-funded summer project at Imperial College London. During these 8 weeks, I joined the Bursary lab to explore the complexities of intracellular plant immune receptor networks. To evade or suppress immune response initiated by cell surface receptors, many pathogens produce effectors. In response, plants synthesize intracellular nucleotide binding and leucine-rich repeat receptors, NLRs, which mediate an immune response in pathogen effectors. Upon recognition of effectors, NLRs are oligomerized into resistosomes with funnel-like structures that insert into the plant cell's plasma or chloroplast membranes to trigger an influx of calcium ions. This executes the hypersensitive response, HR, a type of localized cell death that limits pathogen spread. Some NLRs subfunctionalize and form complex networks, with sensor NLRs detecting effectors and signaling to helper NLRs, which mediate HR. Though NLR networks are well characterized in model plant systems like Nicotiana benthamiana, we lack a firm understanding in a broader range of angiosperms. My summer project aims to explore the NLRs of a novel plant system, the broadbean, Vicia faba. This species could be a gateway to improving our understanding of immunity in the globally important Fabaceae family, guiding future biotech developments for disease resistance in leguminous crops. When arrived at Imperial, the Bursary lab had identified 11 potential NLR genes of clade CCG10 in the broadbean genome. My first step was to use bioinformatic approaches to classify the NLRs into predicted sensor and helper roles. This involved AI-based structural modeling to predict the resistosome structures of the 11 NLRs, as well as construction of amino acid alignments to identify conserved hydrophobic residues associated with HR. The next step was to express the NLRs in plantar. We aimed to transiently transform broadbean plants along the model system Benthi. Observing HR patterns in cell death assays in both plant systems would help reveal the roles of each NLR. We utilized the Golden Gate assembly method to construct level 1 plasmids containing an NLR gene, a GFP gene, and a 35S promoter and terminator. I heat shocked these plasmids into E. coli and harvested them via mini-prep. After checking the plasma purity with the nanodrop machine, then a quick genetic screen, they were ready for transformation into agrobacterium via electroporation. Our transformed agrobacterium could then be infiltrated into plant systems, with cell death assays conducted 3 days post-infiltration. This study represents an exciting first step into improving our understanding of immunity in the Fabaceae family, and I had a brilliant time taking part in this research. I'd like to thank Dr. Tonka Buskert and the Buskert Lab for hosting me during my time at Imperial, and to Freddie King for his day-to-day supervision. I'd also like to thank the BSPP for providing me with the summer vacation bursary that made these past few months possible.